In this video, we're going to be discussing the HMP shunt, which is also called the pentose phosphate pathway. So right here, we're going to have the glycolysis pathway. Like we said before, we're going to start with glucose and we're going to end with pyruvate. Now, the HMP shunt is called a shunt because we're going to shunt glucose 6-phosphate out of the glycolysis pathway, and eventually it can re-enter the glycolysis pathway in the form of fructose 6-phosphate. So when glucose exits the glycolysis pathway and enters the HMP shunt, it's going to go through a series of enzymatic reactions that could eventually lead back to fructose 6-phosphate. Now, why does this happen? This happens in order to produce two molecules that the cell needs. So it can produce NADPH and it can produce ribose 5-phosphate. Now, the NADPH has multiple functions inside the cell that we're going to discuss in a future slide, and the ribose 5-phosphate is used for pyrimidine and purine synthesis that's used in DNA and RNA. Now, NADPH is going to be composed of two nucleotides. One of them is going to be adenosine, which has the nitrogenous base adenine, and the other one is going to be a nucleotide, but it has a nitrogenous base called nicotinamide. Okay, so the HMP reactions can be divided into two groups, the oxidative reactions and the reductive reactions. Now, in the oxidative reactions, we're going to start with the glucose 6-phosphate molecule, and we're going to convert it all the way to ribulose 5-phosphate. Now, this is a series of enzymatic reactions, and in the process, we're going to produce NADPH that could be used by the cell. The reductive reactions happen after the oxidative reactions, and we're going to pick up ribulose 5-phosphate, and we're going to convert it all the way into fructose 6-phosphate. Now, one of the intermediate byproducts is going to be ribose 5-phosphate. Now, this reaction can halt at ribose 5-phosphate, and this ribose 5-phosphate can be used by the cell for purine and pyrimidine synthesis, or it can continue to form fructose 6-phosphate that could re-enter the glycolysis pathway. So now we're going to discuss the oxidative reactions in a bit more detail. So like we said before, we're going to start with the glucose 6-phosphate that comes all the way from the glycolysis pathway, like we said right here. And we're going to convert it to 6-phosphogluconate by the enzyme G6PD, or glucose 6-phosphate dehydrogenase. Now, this is a very important enzyme due to its clinical relevancy, and we're going to talk about this in a few slides. So when glucose 6-phosphate is converted to 6-phosphogluconate, it's going to produce one molecule of NADPH. The 6-phosphogluconate can then be converted into a molecule of ribulose 5-phosphate, and in the process producing another molecule of NADPH, and these two NADPH molecules can be used by the cell for different functions. Now, in the reductive reactions, the ribulose 5-phosphate is going to be converted to ribose 5-phosphate by the enzyme phosphopentose isomerase, and this ribose 5-phosphate can be used for DNA and RNA synthesis. The ribose 5-phosphate can also enter or continue through a series of enzymatic reactions to convert it back into fructose 6-phosphate that could be used in the glycolysis pathway. Now, the enzyme that's responsible for this is going to be transketolase, and it's going to be responsible for the formation of fructose 6-phosphate from ribose 5-phosphate. And of course, it's going to the transketolase enzyme needs a cofactor, which is going to be vitamin B1 or thiamine. So the whole goal of the HMP shunt is going to be the production of two molecules, ribose 5-phosphate and NADPH. Now, the ribose 5-phosphate has the following functions. It's going to help in purine and pyrimidine synthesis that's going to be used in DNA and RNA. And of course, this is vital for any cell. The NADPH has multiple functions. It could be used as a cofactor in fatty acid and steroid synthesis. It helps in the destruction of phagocytose bacteria inside of phagocytes, and it also helps in the protection of cells from oxidative damage. Now, in other modules, we're going to discuss the first two functions of NADPH, but in this video, we're going to discuss how NADPH helps protect cells from oxidative damage. Okay, so how does NADPH help protect cells from oxidative damage? Well, there are certain triggers that could happen inside of our bodies, like infection or certain drugs or inflammation, that could lead to the production of oxygen-free radicals like hydrogen peroxide. Now, so the most vulnerable cells to hydrogen peroxide or oxygen-free radicals are going to be our RBCs. So it's very important that our cells have a mechanism in order to neutralize these oxygen-free radicals. Now, the way we neutralize, or our cells neutralize these oxygen-free radicals is by glutathione. So glutathione is going to react with hydrogen peroxide, and it's going to produce water and glutathione disulfide, catalyzed by the enzyme glutathione peroxidase. Now, the glutathione disulfide is then going to be regenerated back into glutathione by the enzyme glutathione reductase, and it's going to consume one NADPH molecule along the way. So we can conclude that in order for our red blood cells to resist oxidative damage, by hydrogen peroxide, we need to maintain adequate levels of glutathione. And in order to maintain adequate levels of glutathione, we need to maintain adequate levels of NADPH. Now, the source of NADPH is going to be from the HMP shunt. So in order to maintain adequate levels of NADPH, we need to have a fully functioning HMP shunt. So this brings us to the pathology that could happen in the HMP shunt, and it's called G6PD deficiency, or glucose 6-phosphate dehydrogenase deficiency. Now, this is the most common enzymatic deficiency that could happen in humans. So this is an X-linked disease in which these patients have a deficiency in the enzyme G6PD. And of course, this is going to stop the whole HMP shunt pathway, and it's going to prevent the synthesis of NADPH. So if we cannot regenerate our NADPH, that means that we cannot regenerate glutathione, and we're going to be very vulnerable to hydrogen peroxide inside of our RBCs. So the RBCs are going to then accumulate hydrogen peroxide, and that's eventually going to lead to hemolysis of that RBC. And this is characteristic of G6PD deficiency, in which there is intravascular hemolysis of RBCs.